Hello and welcome to another episode of Breaking Bio. I'm Tom Housley and I'll be your host for this episode, which promises to be magical because we have Professor Hope Jahren joining us today. Hope is the writer of the excellent Hope Jahren Sure Can Write blog and also curator of one of the most hilarious and consistently informative Twitter feeds in existence. But not only that, she's also a professor at the School of Ocean and Earth Science and Technology at the University of Hawaii, with a list of awards and honours that would probably take me the entire episode to read through. But rest assured, she's a big deal, not just on the internet, but also in real life. And Hope is joining me today from New York, so thanks for coming on Breaking Bio, and maybe we can start by you giving us a quick overview of your research.
Thanks. That was quite an introduction. Are you sure you're talking about me? Yeah. So I'm Hope Jahren. I'm traveling, so this is my incredibly fancy hotel room in the city. It's almost big enough for me to turn like 360 degrees when I'm standing, but not quite, so I can do like a 270. I'm going to be broadcasting today from this hotel room. So you might hear some street noise because the police might be coming for me any moment. It's loud here in New York City. What else do I do besides travel? I travel a lot, but I also study plants. Plants kind of as a concept, more than as actual plants, in that I don't know a lot of species of plants. I can kind of tell the difference between an oak tree and a pine tree, for example, with pretty good accuracy. But I am more interested in the way plants work biochemically, and how they have been so successful for so very long, hundreds of millions of years, and what might happen in terms of the fact that their environment, their resource base is changing very quickly right now, particularly in the last even 50 years. And that's got big implications for forestry, for agriculture, and just for understanding the types of metabolisms that the Earth has made home for.
So how do you actually study what plants were doing in the past?
I study it by sitting in my office and writing a lot of grant proposals. And when I'm not doing that, I sit in my office and I revise other grant proposals. And when I'm not doing that, I review other people's grant proposals, and that has somehow become a huge portion of what I actually do, and it promises to be even more in the future. Back when I used to be able to be in the same room with like a plant instead of a computer, we would do this by growing plants under very controlled conditions. So we have these little space-age kind of chambers where we can monitor absolutely everything they're exposed to. We can increase and decrease the relative humidity at will and light levels, and importantly CO2 levels, because CO2 change is a huge factor in what's happening today. You read about it on the news every day, and also it's a fundamental resource that is varied by several orders of magnitude in its availability for plants over hundreds of millions of years, which has, we believe, strong implications for how they've evolved and what they might be able to tolerate, and the myriad of reactions and compensations that might arise as things change very quickly nowadays.
So are you looking at kind of predicted atmospheric changes and how plants will react to this, and what kind of things have you found?
So the interesting thing for us is that I'm not the first person to look at this. So CO2 is a basic resource for all plant growth, and CO2 levels are changing, and they're predicted to change. There's in my eyes, there's no way that CO2 levels aren't going to keep going up by quite a bit as people in, China and India acquire cars and acquire electrical capabilities in hospitals and for street lights and all this kind of business. So there's going to be more fuel burned in the near future, I believe, and CO2 levels. I personally believe we're going to see 1,800 ppm before our grandchildren are sort of done with their time on the planet. So I'm interested in the physiological responses of plants through the trajectory that extends from today up into these very high levels. There's a lot of previous work that's been done on this. Most of it centered on nailing down responses at 550 ppm, which was a number that the IPCC put forward in the early 90s as the likely scenario for 2050. And it created a kind of rallying point for biologists and physiologists to study. The interesting thing, since the early 90s is, we're going to blow past that number probably earlier than 2050. And it has become interesting to think about, what physiological responses might be at much higher levels. The reason we got interested in this is that I'm trained as a geologist and a soil scientist, and the moment at which there were some key innovations in plant life were also periods of extremely high CO2. We're not sure exactly how high, but maybe 14 times as high as today. And so for those reasons, it became exciting to work at those very high levels, 3 times, 4 times, 10 times what the ecologists were doing. And when we did that, we started to see very different responses than what you might predict from just extrapolating beyond that 550 point that people studied so furiously.
So you said that these levels were really high in the past. So how far back are we talking?
Right. Okay. So there's interesting things about how life has developed on land. Plants, as you know them, trees and bushes and weeds that you pull and all that kind of business, that stuff evolved, somewhere around 450 million years ago was land first colonized, right, from, more or less bare rock. That's interesting because land was, kind of sterile while the ocean was teeming with life. And so one thing I try to impress upon people is that the history of life and evolution on land is very, very separate from what happened in the ocean, particularly for plants. So again, those plants, the trees and bushes and the stuff you've got in your garden is far more closely related to you than it is to the phytoplankton, the tiny little plants that are floating around in the ocean. So where am I going with this? So land is an interesting thing to study. It's also interesting because plant strategies have sort of been accumulating. We don't have, we have some stuff that's gone extinct, but for the most part, plants, there's just more and more of them all the time. So land is kind of still filling up. We don't have these big absences from the animal record. We have ferns back then, we have ferns today. We have conifers developed, plants without flowers. We have lots of those today. And then flowers hit the scene, and now everything you see almost is a flower, but there's still kind of room for all these forms that existed before that. So the trajectory of evolution on land is really interesting, and as you noted, there are some very key moments. So one key moment is, the colonization. What were the first plants to take hold, and sort of how did that happen? About 450 million years ago. Very soon after, trees developed, so woody structures that could climb really, really high. One problem with being on land as opposed to the ocean is that you can't float and it's not wet. So plants on land have had to develop um, interesting strategies toward both getting water down low and getting light up high. One strategy is to stretch. And if you're going to stretch, you've got to deal with gravity. And wood became an amazing innovation in terms of, being stiff and getting up. Gosh, what's a taller organism on land than a tree? Who can do that, right? So that happened fairly early on. Wood is just something that plants can biochemically do. Some bushes, if you've ever dealt with poison ivy, you know that it can be very rubbery, but if it gets a really good place that it likes, it can actually supplement its tissues with wood, and kind of turn into this really hardcore form of poison ivy, which is much more structurally sort of sound. And so there are these interesting innovations that kind of cut across taphonomies or phylogenies. Not taphonomies, phylogenies. So I'm interested in how those are kind of triggered by the environment. Perhaps one of the neatest things that we study is there was a critical period of time in the early Cretaceous. So this was in the time when dinosaurs were really diverse and the continents, Pangea was kind of breaking up, and flowers evolved. And once flowers evolved, they very, very quickly almost took over the landscape. Before 125 million years ago, there weren't, a lot of flowers around. It's hard to find the very first fossil incidence of something, right? So we tend to find fossils once things, are reasonably common on the landscape. But having hope of finding the very first incidence is tougher. So 125 years ago, flowers kind of got going, and within a very brief time span, 5 million, 10 million years, they had proliferated and were extremely successful. Almost everything, you look out your window, and unless you're in the conifer forests of the high latitudes, almost everything you see out your window is some kind of flowering plant. Even if, grasses, the flowers are very, very little, but they're there. So the interesting thing to us about that time period, it was also a time period that we believe of extremely high CO2. And that's pretty well documented both in proxies that allow us to estimate what those CO2 levels were, but also in the fact that there's massive volcanic eruptions. There's these big basalt lava flows. Basically, a lot of the western U.S. and most of India is a huge lava flow from that time, and a lot of CO2 comes out of volcanoes. So this great moment when there was a revolution in plant reproductive strategy is also a moment of very high CO2. So I was interested in trying to put these things together in terms of the role that might have played. I'm going somewhere with this. And then we became somebody that was interested in these very high levels that are beyond this sort of near-term estimate that the IPCC had made. So we're in this interesting position of looking at not 2050, but 2300 sort of projections for the future.
That's very cool. So what kind of techniques are you actually using to kind of go back and look at what was happening in these ancient times and figure out how they were affecting the proliferation of reproductive strategies and what kind of species there were there?
Right. So so again, I'm interested in biochemistry and physiology that cuts across all plant organisms, basically. So it's sort of like not studying a species, but studying, if I were studying animals, I'd be studying breathing, for example.
Okay.
So trying to study processes that are germane across taxonomies, both living and both living now and living then. So, so what we do is we grow Arabidopsis, which is this little kind of representative plant that the geneticists like to work on because the whole genome was known very early on. So we put it in, you know, kind of a big plastic box, and we modulate every environmental attribute you could think of, and we study its growth, and then we harvest the tissues and measure the tissues in a whole bunch of different ways. We can do genetic work on it, so we can look for genes being turned on and off, and we know those are genes that all plants share. So there are you know, strategies for extrapolating what's going on in this one little guy to you know, how a ginkgo tree might have reacted, etc. And I also use stable isotopes, which is a nice way of following how carbon kind of gets passed around. Because the extra neutron in heavy carbon can serve as you know, basically a tracer, and the sorting of those atoms is different at different states in the, you know, biochemical machinery, and so we use that as kind of a clue as to, you know, the balance between photosynthesis and respiration, for example, and a way to gain insight into that.
I want to come back to the stable isotope stuff in a minute, but I was reading before about some of the work that you've been doing on Axel Island.
Yeah.
So this is somewhere that looks very different today than it did in, I think it's the Eocene era that you were looking at. Can you tell us a little bit about this and the work that you were doing there?
Okay, so this is in the high Arctic. It's about 1,000 miles north of the north coast of Alaska, and it's in Canada. And the thing that's interesting about it, well, there's several things that are interesting about it, but for us, what's interesting about it is you can find just massive amounts of plant material, wood, etc., you know, logs, and this stuff is incredibly well preserved. It looks like, you know, driftwood from the beach. Actually got in some trouble on the media because I said something like, you know, if I didn't know better, I would think this was, you know, modern driftwood from the beach. And of course, folks that don't believe that the Earth is very old kind of had a field day with that. But it's extremely well preserved, and it is Eocene in age, which puts it at about 45 million years ago. And it's located well above the Arctic Circle, and it was located well above the Arctic Circle. And it's cold up there now, and so we have these lush forests. I mean, some of these trees are 3 meters in diameter. It's not a marginal ecosystem. This is a lush, thriving ecosystem. And you look up there today, and you know, you don't have you have like lichens and stuff. You don't have trees up there today. So it was warmer than it is today. You know, the climate has changed, etc. But one thing that hasn't changed at those latitudes is that there was 3 months of darkness every year and 3 months of continuous light. And both of those are very stressful realities to a photosynthetic organism. I mean, you can try that with your houseplants at home. Put them in a room for the dark for 3 continuous months and see how they fare. Or put them in the continuous light for 3 months, and that's also, you know, if I kept you awake for 3 months straight, you would probably be pretty ragged at the end of it. So how do you get a forest that can not just survive those conditions but thrive through them? And those forests, we believe, persisted for, you know, 10 million years. And this was not a freak kind of episode in Earth's history. This was a long, stable configuration for planet Earth. So it's, you know, it's akin to, you know, finding humans that could breathe underwater. You know, this is really a compelling moment in terms of our plants doing something during this period that they're no longer capable of, or are plants today capable of this, but they no longer invoke it, you know, is one of our questions. And in fact, part of my research group is in Siberia right now, because it turns out that these forests were circumpolar, which means that they existed above the Arctic Circle all the way around the globe. And so finding these fossils, and particularly finding examples where they're very well preserved, is something we've been working on for 15 years now. But
um,
that's a great problem in terms of just knowing how plants work on planet Earth.
yeah, yeah. So, do you think there's any chance of this kind of uh, yeah, the ability of plants to survive in that kind of environment is dormant, or whether this is completely disappeared, or I guess that's maybe what you're trying to figure out.
Yeah, I mean, I have theories about this. I, I mean, I think anywhere where you don't have permafrost, you can have a forest, is one of my theories. And that it's actually the fact that the water that you need to be a plant is locked in this frozen state, and is thus inaccessible because it's solid. I think that may be, the crucial control about where you have stuff, and not light, which is a little bit amazing because you need light to turn on photosynthesis. So you think of light as entering the system upstream of where water does. But, you know, uh, if you can get liquid water, you can make a go of it, is one thing that we're toying with. And certainly if you take a road trip and you drive across the United States, you'll convince yourself very early on that where it rains, you see plants, and where it doesn't, you don't. And so the importance of water is something that I think an awful lot about nowadays.
Okay, so I want to switch slightly from plants across to humans, because I was reading that your work with stable isotopes has led you to slightly new directions for investigating links between human blood and diet. Can you tell me a bit more about that?
Right, so all the sugar that you've ever eaten originally comes from a plant, right? So, and your brain needs sugar to keep you going. And essentially, plants are the only organisms on Earth that can make inorganic carbon into sugar. And then everything besides plants are just, parasites that evolved to feed off that sugar. And so I've thought a lot about, how plants make sugar, what the resultant chemistry of those sugars are, how the isotopes partition into those sugars. We've also got this interesting situation now where people are dosing themselves with incredible amounts of sugar. And these plant sugars then, it's almost like a tracer, right? So you've got this diet where you just pulse in this huge amount of sugar. And because these are plant sugars that we understand their isotope chemistry, we can kind of follow this around in the human body. And it has the potential to make really interesting contributions to dietary analysis. I work with some nutritionists that study people. They have experiments. They have feeding studies where they invite subjects to take part in controlled feeding studies where food is chosen and the amount that's ingested is carefully cataloged, etc. And one thing that we've talked about is how it doesn't so much matter what your last meal was in terms of diet. It matters, how frequently you're eating dessert with your meal, or what your long-term habits are. And it's surprisingly hard to get good data about that. If I asked you, you might be the most compliant subject ever, but if I gave you a pencil and said, write down everything you've eaten in the last 40 days, you're going to give me very bad data no matter how hard you try. And so finding some kind of, we call it bioassay or a biomarker for how much sugar people have eaten, over a 40-day period would be really super useful to people that are trying to link sugar consumption with various health outcomes. And so we're hoping to, we're collaborating with nutritionists and hoping to contribute to that problem.
eliminating the unreliability of humans sounds like a good idea.
Yeah, yeah, I mean, again, and then if you ask people to write down everything they eat for 40 days, they will invariably come up with a very strange sort of intake for those 40 days. It really affects what people end up eating. So some kind of measure that you can come in after the fact, and hopefully, you could get this out of a blood draw that was done for other reasons, etc. Give people information about, the dream scenarios that you could come up with something like how they do for cholesterol, where you go to the doctor and you say, I'm having these symptoms, and they do a lab test and say, well, your cholesterol is this number, and I'd like to help you get it down to below this number. And if we could do that with sugar, it would clarify approaches to diabetes and other kinds of diseases that are much squishier in terms of, well, how much soda do you drink, and this kind of stuff. If we had kind of a target that people could work with pragmatically, that would be a huge contribution to how we manage some of those diseases.
Okay, I want to just finish by with something a little lighter. So you kind of exploded onto the social media scene some short time ago with your blog and your Twitter, and I was just wondering how you've found using social media has helped in terms of your research and collaborations and things like this, and what have been your highs and lows?
Yeah, it's kind of an outlet for me. My blog is almost a year old, and it's just a lot of fun. It gives me a place to say things that that the people who know me well are tired of hearing, and so I need new people to say those things too. And some of it's helpful. I mean, one thing I try to do is provide pragmatic advice for what I believe is how to do this stuff right. How to write a grant that actually has a chance, and how to get yourself through these interview situations, and how to deal with situations that are fundamentally unfair sometimes. And it's it's meant to be useful. It's my voice, the way I actually am. It's what I actually sound like, around my friends and stuff like that. And you don't get to use that voice when you write proposals and scientific papers. And so it's been a great, joy and a great outlet to find a place for that kind of expression. And so, honestly, it's a very selfish thing. It lets me release a side of myself that I really like. It's a very imperfect side of myself, and I say stupid things, and I make mistakes and things like that. But sometimes I get a real laugh, and that makes me happy. It's very gratifying to me, and it's really just a really social thing, and I have the luxury of creating it, and I've had the incredible gratification of finding a few people that like it, that think it's more fun to read my blog than the billion other things you could be doing. Slightly better than brushing your teeth, or whatever it is. So it makes me happy, and I don't know if it's more complicated than that. I wish I could say that I see this as a very serious form of science outreach. You need to start doing now in the 20 whatever century, and but I like most of the things I do, I'm just indulging myself.
I think it's quite nice to see someone who's not taking it seriously and is having fun, and that kind of shows in all your posts.
It's not serious except for when it is, except for when we're talking about things that I just feel incredibly passionately about, that these subjects we dance around, sexual harassment, sexual assault, just basic unfairness in the workplace. And I am sick of not talking about these things, and that's also why I blog about it. And I have some job security, and so for now, I have some job security, and so it's a conscious choice to kind of spend that capital in a way that also allows me for more, I fancy myself as a bit of an artist, so it allows me for more artistic expression than I've had access to through more pedestrian execution of my job, shall I say.
Yeah, I think it's very inspiring for a lot of people to have someone who's in your position, a professor with some security, to be able to speak openly.
Yeah, it gets me into trouble sometimes, and I have yet to figure out just how much trouble it's ever going to get me into. But, I kind of am stuck being me. I've been trying to find a way around that for a long time, but, as I get older, I'm kind of starting to accept the way I am. And a lot of people have found the internet as a good place to be who they are, and maybe I'm experimenting with that in some way.
Okay, well, I think we're just about out of time, but maybe if you can quickly give us the links to where people can find your blog and your Twitter and see this side of you as well.
Right. So if you punch my name in, I'm pretty. I promote this stuff. I actually write this stuff so that people will read it. So I'm not at all shy trying to get people to read it. Basically, if you just Google my name, which is Hope Jahren, Jahren, that's going to lead you to my blog, which has the words Hope Jahren in the title, and my Twitter handle, which is the words Hope Jahren, and my lab page, which is the word Jahren. So all roads lead to me.
It's one of the good things about having a unique name, which I also know well about. Okay, so that's all for this week. As usual, you can find us at Breaking Bio on Twitter or our YouTube or Facebook pages. Thanks for listening, and thanks again to Hope Jahren for joining us today.
You're welcome. It was great.
Thank you, and goodbye.